Of all the molecular mechanisms connecting nutrition to cellular aging, the ND+/SIRT1 axis has attracted more longevity research investment and more venture capital than perhaps any other pathway in the past decade. The compound interest in this system is justified by the convergence of multiple lines of evidence pointing to the same conclusion. ND+ levels decline substantially with aging, by approximately 50% between ages 40 and 60. And this decline reduces the activity of SIRT1 and related sirtuins that control autophagy, DNA repair, mitochondrial biogenesis, and metabolic regulation. Restoring ND+ levels through niacin and its metabolic relatives appears to partially reverse these age-related deficits in model organisms, and the human clinical data is beginning to follow. SRT1's role in autophagy operates at the level of protein deacetylation. It removes acetyl groups from ATG5, ATG7, and LC3, converting these autophagy machinery proteins from their inactive acetylated forms to active ones. When NAD+ levels are high, SRT1 is active and autophagy runs at higher baseline rates. When NAD+ declines with aging, SRT1 activity falls correspondingly, autophagy slows, damaged cellular components accumulate, and the downstream effects manifest as the hallmarks of biological aging. The SRT1 activation pattern also enhances mitochondrial biogenesis through PGC1 alpha deacetylation, meaning that after mitophagy clears damaged mitochondria, adequate ND+ via SIRT1 promotes the synthesis of new, healthy mitochondria to replace them. This sequential autophagy then biogenesis program is one of the most compelling models for how metabolic renewal could slow biological aging trajectories. Food sources of niacin are broadly distributed across protein-containing foods. Liver leads at approximately 14.9 milligrams per 3-ounce serving, followed by chicken breast at 8.9 milligrams, tuna at 8.6 milligrams, turkey at 10 milligrams, and peanuts at 4.2 milligrams per ounce. Mushrooms provide approximately 2.5 milligrams per cup, modest, but meaningful for plant-based diets. The RDA of 14 to 16 milligrams daily is achievable through ordinary omnivorous diets, but the NAD+ raising effects studied for autophagy and aging benefits require substantially higher niacin precursor delivery than dietary niacin alone typically provides. This is where nicotinamide riboside (NR) and nicotinamide mononucleotide (NMN) enter the picture as direct NAD+ precursors that have been studied at doses ranging from 100 to 1,000 milligrams daily in human trials. Both compounds demonstrably raise blood NAD+ levels in these trials. Whether that elevation translates into the autophagy and aging benefits observed in model organisms is a question that current human evidence is approaching but has not yet definitively answered. The honest scientific position acknowledges the mechanistic plausibility, the compelling animal data, and the encouraging but preliminary human clinical results rather than either dismissing or overselling the therapeutic case. Regular niacin at supplemental doses above 50 milligrams produces the well-known niacin flush, prostaglandin-mediated skin reddening and tingling that is harmless, but uncomfortable enough to cause discontinuation. This does not occur with NR, NMN, or nicotinamide. However, combining niacin and its precursors with fasting protocols, which independently raise ND+ by shifting the NADH to NAD+ ratio, produces the most synergistic autophagy activation available from non-pharmacological nutrition interventions. Liver function monitoring is medically appropriate at high niacin doses, and resveratrol from grapes and berries may amplify SIRT1 activity alongside adequate ND+ in a complementary dietary pairing including in a comprehensive autophagy nutrition strategy.